Bismillahirrahmanirrahim. I am Muhammad Anowar Hossain, Professor, Department of Biochemistry and Molecular Biology, University of Rajshahi. This is a bioinformatics tutorial. Today, I would like to talk about RAST, Rapid Annotation using Subsystem technology. So this is an bioinformatics tutorial for genomic analysis, or particularly bacterial genomics. Let's see why RAST annotation server. It is an online server for functional genomic analysis. And if you want to annotate your bacterial genome or near assembly bacterial genomes, you can use this server for rapid annotation. It provides high quality genome annotation for the genome across the whole phylogenetic tree. You can use the result of RAST genome in the form of seed view for the preparation of manuscript in the journal submission. You can get subsystem categories and subsystem features of the genome. And you can also compare the metabolic reconstruction of this organism, that of the another organism. Just you can compare both, and you can find out some difference in their metabolic function, metabolic pathways. And you can get the comparison between the function and sequence via cake metabolic function observer. What is seed? Seed actually it's generating the hash values of short sequences called seeds that enables quickly identifying similarities between the genomic sequences by matching seeds with a single lookup of their hash values. You can see the RAST that accurately annotate the genomes, and this is submitted the subsystems technology, and which curated the subsystems, then fixed technology used to just finding the similarity of some iso function, the genes, and then that it curated in the fixed forms, and so that called again RAST show you the result. So when you use the RAST, the seed view form of the bacterial genome you have analyzed, you can get the result this like the say pie chart. Here the subsystem coverage, and this subsystem coverage, this 40% means the 40% the gene identified in this system RAST technology by the RAST technology, they are deposited in the software and the web server, and the 60% gene of this bacteria are just not specific, that just they put for the some protein, but that the protein function is not yet known. So, and you can find the different color in the pie chart, the subsystem category distribution, and what are the function and what type of gene are involved in the what type of metabolic function in the bacteria. So now let's start how to use RAST. So at first we have to go to the Google web server, and so this is the Google page. Just we can write the Google search, Google search, and we can write in the Google, just ask the Google to the RAST genome server, RAST annotation server. Just click there. You can see here the already login. So I already log in this website already. So if you don't have account, just open here, and then just from the you have to log off using your email address and your name or username. Then it is free for all the scientists and students. So you can first log off and then log in. You can see here, I already log in, Muhammad Anwar Hossain. So now we can upload a job. You can see I already, this is the RAST website. You see Rapid Annotation using Subsystem technology is a fully automated service for annotated complete and nearly complete bacterial or archaeal genomes. It provides high quality genome annotation for the genome across the whole phylogenetic tree. So here we can upload just upload new job. This is the new job. Just please wait. It's moving. So upload a genome. This is upload a genome. So you have to have a raw genome or semi-assembled annotated genomes. So here we can download some bacterial genomes from the genome website NCBI, and then we can use this as template. So from here we go to another website, just NCBI nucleotide. So here is the NCBI nucleotide, and we can Bacillus cereus. You can see Bacillus cereus. So this is the Bacillus cereus complete genome sequence here. You can see Bacillus complete genome sequence. Click. From here, you can download from the sequence, the file, the gene bank, file format gene bank, the gene bank format, and create the file. Just wait few seconds, then you can find the genome sequence here in the gene bank format. Okay, maybe this already completed. So this one is the sequence 2, genome sequence. We can see what type of genome downloaded here. Open with the notepad. You can see Bacillus cereus genomic DNA, complete DNA. You can see the annotated gene bank format. You can see the coding sequence, and also finally in the last part here, in the last part, you can see the DNA sequence. So this is the gene bank format. You can see the gene bank format. So always after downloading the sequence, you have to check whether it is correct or not. So you can see this on the sequence. So now we can upload this sequence to our RAST server. Is the RAST server. So from here, RAST server, you see choose the file, the download. Where is the download? This download, and the sequence 2. You see, okay. Then use the data to go next step. You can see that it's moving. See, upload a genome, the review genome data. So this on the already this is RAST web server. We have analyzed your upload and have computed the following information. So here already got all those things. They already uploaded our sample in their website, and now you can see taxonomic ID. This is also okay, and this bacteria Bacillus cereus, and Bacillus cereus 401. So Bacillus and Cereus. So this is the strain number. Everything is okay. And the genetic code we use the 11 archaea, most bacteria, most viri, and some mitochondria. So we have to choose. We use the default system to go to the next steps. So go to the. I use this data to go to the step 3. So okay, now complete the upload, and now you can see the rustic, or you can use the classic. So first I can use as it is, and so automatic fix the errors, and so nothing, all the just finish the upload. So we can see the view the job status. Job is working. The server is working, right? So this one is the already we submitted this the working. The server is working on our Bacillus cereus and our annotation scheme RAST TK. So rapid propagation has started, quick check has not started, quick revision started. So all those things will be done. So still now it's waiting. It is waiting. So it takes little bit time. So we have to wait for that and share this genome with the selected user. So here from back to the job overview. So we can use another one, say new job. We can go for the new job. So from here, for the classic selection, we can use the same on. Okay, get to the next steps. We have to use the same bacterial genome. Okay, everything is the similar. So go to the next steps. Okay, so this time we use the classic RAST. We use the classic RAST. Okay, so everything is okay. Not necessary to change. All are default here. So just finish the upload. So now we already have submitted two job, and let's see what is the job status here again. This one is the 75, last 75. This one is also is working, and we can see our submitted job overview here. You can see this today I submitted this job 74 and 75 serial. So this one is just not started. This is not started. So we have to wait for a long time, and maybe it takes little bit time. So starter job load is very heavy. So we already submitted to the job, and both are the Bacillus cereus NC strain NC740 So just wait. You can see here. So many people are working on the bacterial genomics. As of Monday, September, you see today there are 719 jobs in the Rust GUI. So very heavy load on this server. Let's wait. There is still now no progress. Okay. Just again, job overview. Okay, we can see our job is already completed now. Both are already completed. So let's see view the details. So first on, we can see this one is the 74. So what we see here, the genome ID here, and we use RAST-TK. So this already all is finished successfully. The number of features is 5,542. Number of fatal problem, no problem, no earnings. So everything is now is complete. So now we can download the gene bank from the here. So the after the download file, and also we can, if we want to publish this genome annotation, the gene function. So we can see the seed viewer. You can see now just coming. You can see here the seed view, the subsystems statistics here 24% in this uh, server. So 25% is the protein has the similarity with this bacteria, and 76% is the unknown hypothetical protein. You know, the non hypothetical protein and hypothetical. So these are not in the subsystem, but these are in the subsystem. So subsystem only 24%, 76% is non subsystem. So they are not in the server, but they have the sequence, the coding sequence that encode the protein, but they are not available in the subsystem. So you can see here the carbohydrate metabolism, the red in the metabolic nucleotides, and this one is the amino acid derivatives, and there's the protein metabolism. This one is the fatty acid metabolism, respiration, cell wall, so and their iron acquisition, metabolism. You can see miscellaneous and also regulation cell signal in potassium metabolism. So and also the many the DNA metabolism, nitrogen metabolism, the dormancy correlation, and also you see the carbohydrate metabolism and metabolism, aromatic aminic acid. So all the uh, feature, the subsystems, what are the function of protein annotated, you can use this and as the publication, and also you can compare, you can download from here, you can see, you can download the picture, and also you can just see the feature of the subsystems, the what are the uh, um, um, genes involved in the different type of pathways. You can see the metabolism aromatic acid. So you can choose from the what type of the say carbohydrate. We choose the carbohydrate metabolism. So in the carbohydrate metabolism, what are the gene? The 192 gene are associated with the carbohydrate metabolism in this bacteria. So serious, Bacillus cereus, and also you can choose from here, say the first all, and then you can choose another one, say uh, virulence disease, or stress related something. You said stress response protein. You see the oxidative stress, stress containing proteins. You see the stress, glutathione, antioxidant space. So these are the protein you can find out, the stress responsive gene, and also you can find out, say uh, say the what are the virulence or disease defense proteins, resistance, antibiotic, toxic compound, virulence. So all the, you see the streptomycin resistance, streptozotocin resistance, tetracycline resistance, or all the resistance gene. What are the resistance, multi-drug resistance gene, or the cobalt cadmium resistance gene? So these are the bacteria, these bacteria resistance to the cobalt, zinc, cadmium resistance. So all those things you can find out the gene function. So for the correct annotation and the publication, so you can use this type of subsystem information annotation results. And also if you want to compare the, say you can compare here, the compare with the metabolic function via cake, then you can click here, the say you can compare with other related bacteria. You can see here, say you can we have Bacillus cereus, right? So we can compare with the Bacillus serious other strains, say other strains. You can see other strain, or Bacillus related, the Bacillus cereus harbor. You can, okay, see, uh, E333. Okay, this just select. Okay, so if we select Bacillus NC7401 and Bacillus cereus E3, two strains, we can see what about the gene involved in the stress response. We can see the stress response. You can see the A and B has the cold shock, the protein, right? Yeah, B has, yes, that means they have the cold shock or heat shock protein. B has heat shock protein, but in case of A, the find no. So it has no stress responsive the protein, heat shock protein. So that these are the difference. So you can compare with the two bacteria genomes with the and also you can use carbohydrate metabolism. Say this carbohydrate metabolism. Carbohydrate metabolism. Okay. So in case of carbohydrate metabolism, you also see the similar. So Bacillus cereus and the Bacillus thuringiensis cerebor and Tromocydas has a similar carbohydrate metabolism. And next we can see more that because 193 gene is involved, you can see this all are the yes. That means the similar. So we have to go to our job overview. We can now see the what about the our 75 number. So we see the 75 job, number 75 job. Okay, this one is we choose the different annotation script, just like classic RAST. So classic RAST, you can see 5,494 gene, the number of gene and all those things are no problem. So here we can see the seed view. You can see the seed view, the cofactor vitamin for prosthetic. So the carbohydrate metabolism, this and the amino acid derivatives, and this one is the RNA metabolism. So from here we can compare again you see it is the 41%. 41% has the in some gene the similarity in subsystems and total 2,174 gene, the non-hypothetical 2,062 and hypothetical 112 and 59% in not in subsystem. So they don't have the similarity with the our the server RAST server. So this protein has this bacteria has some protein. They input some gene, but they have no similarity with the subsystem. And you can also compare and download the picture from here, and also uh, compare with the other uh, mm, uh, bacteria. You can see the compare with the metabolic reconstruct Bacillus cereus on with the other bacteria, say any another Bacillus, Bacillus cereus or Bacillus subtilis, say Bacillus subtilis. You can see Bacillus subtilis. Select, then we can see the comparison of the both two Bacillus cereus and Bacillus subtilis. What are the difference? What are the metabolic function difference? You can see easily the all those things Bacillus group. So most of the gene are very similar, and also we can check here what is the difference. Say first one is the amino acid derivatives. We can see what difference the amino acid derivatives. So we see the amino acid derivatives. There is such big difference, and amino acid derivatives, and also next we can see what is the difference, no difference, and no difference. and no difference. So amino acid difference is not so significant, and you can see the difference in the last one. You can see the difference here. You can see the amino acid derivatives, some difference, proline hydroxyproline uptake and utilization. So fine in the yes, that means the Bacillus cereus, it has the gene, but subtlety has no gene for the amino acid derivatives, UC derivative for proline and hydroxyproline. So there is no gene. So you can see the no. And also here is also amino derivative proline hydroxylin and proline 5-carboxy reductase, pro-glyc protein, it has no. So, and the, but subtlety has this type of gene. So by this way, we can identify the difference of the genome to genome difference and what the metabolic functional difference, and we can also use the some cake pathways from here, and we can see that the metabolic pathway also. So let's go back and see the seed viewer. You can see the subsystem feature, all the gene responsible for that. If you want to see any type of gene, say potassium metabolism gene, we can see here what all the gene are involved in the potassium metabolism. Okay, so by this way, you can use and prepare your publication, and also you click here, there is the annotate. So you have already enabled editing for the other private genomes, so but not here for this one. You can enable the annotation capabilities for the genome here. So from here you can annotate all the genes for annotation capabilities of this seed viewer. From here you just activate the genome and we enable, but not necessary, because already these both bacteria we received from the internet, so these are not novel, already discovered. So when we have new, then you can use this annotation server for the annotation. And so by this way, we use this server for genome functional genome annotation, and so on, so many things we can do. So if you like this video, you can share this video to others, colleagues, researchers, and also don't forget to subscribe our channels so that you can get more updated video regarding the bioinformatics and genomic analysis and all the bioinformatics related video. Thank you very much.